So I'm a bioengineer, and I've spent most of my professional life trying to understand how enzymes, which are the catalysts of chemical reactions inside cells, work, and especially how they might be regulated by metabolites or other proteins. And I'd love to talk to you about that. But instead, because we're talking about botany, I want to talk about a project that we started about half a dozen years ago, which really grabbed my attention. It has to do with developing next-generation plants for use as biofuel feedstocks. So why plants? Well, we can make biofuel from plants because they're great sources of cellulose. So many of you know cellulose is a polymer of glucose units that are strung together. It's in fact the most abundant polymer on the planet. We can deconstruct that polymer into glucose units and ferment that into ethanol, which we can add to our gasoline and extend our fuel supply until we can convert to some sort of hydrogen economy or something along those lines. So lots of plants are being considered to be used for biofuel. A lot of specialized plants, including some crops, which a lot of people aren't too excited about. But I think a reasonable question is, can we really use plants to meet an ambitious goal of replacing nearly half our gasoline by 2030 in the United States? Well, if we were able to manipulate some of the traits of those plants, some people think we might be able to start to farm plants, like in this case, poplar trees, to be able to harvest for use to convert that cellulose into ethanol to extend our fossil fuel supply. So tonight what I want to tell you a little bit is about my motivation for trying to get involved in this program. I am not a botanist, all right? I'm an engineer, biochemist and engineer. What some of the challenges are, what we're trying to do to address those challenges scientifically, and how I've only been able to do this through interacting with a wide variety of experts, colleagues, friends, scientists, including the general public, like tonight. So, what got me motivated? Well, about 10 years ago, I was part of a leadership group in the state of Maryland that was looking at a variety of problems in the state. And one of the problems we talked about was transportation. And if any of you had to drive here, you know exactly what I'm talking about. There's that problem, but there's also the problem about fuel for all the cars that we drive. And the question was, around that time, around 2008, some of you might remember, certainly some of you are old enough to remember, that the price of crude oil was high, over $150 a barrel, and gasoline in California was well above $5 a gallon, where I was living at that time. So also around that time, a friend of mine gave me this book by Paul Roberts. It's called The End of Oil. It was a fascinating book, page-turner for me, and it really transformed my thinking, because what Roberts' thesis was is he thought that all the oil-producing countries in the planet were overestimating the oil reserves that they had. And he predicted a much earlier shortfall than a lot of experts and a lot of three-letter agencies like to think we have in store. So that really got me to think, what would life be like without oil? Think about it for a minute. No gasoline for our cars, that's easy, all right? But no lubrication for any machinery, no dyes, paints, textile, materials, a lot of different materials. It would be a very different world, very interesting. And I thought, well, maybe there's something to this problem that I could apply some of my expertise to. And around that time, I'd also built a building for the University of Maryland that had a state-of-the-art plant laboratory in it, plant transformation lab. And my Boston University president said, gee, Ed, there's not nearly enough activity in this lab. What could you do about this? And I thought, well, maybe I could take this opportunity with these new labs, with this interest in trying to address this energy problem by working on plants to maybe make biofuel. So how do we make fuel from plants? Well, I alluded to the fact that plants contain cellulose, and a great source of cellulose is wood. Wood's about 75% cellulose. The other 25% is another polymer called lignin. And lignin is a phenolic-based polymer. It's a chemical that it's the second most abundant polymer on the planet. And it has a lot of functions in plants that are important. It provides a lot of tensile strength. It makes wood the great material to build a lectern like this or the chairs we're sitting on, a lot of that frames for houses we live in. It also helps line the vasculature in plants so that it can more readily transport water. Provides that structure for growing trees. It also has an effect as a sunscreen. So plants are out in the sunlight all day. They suffer the effects of ultraviolet radiation from the sun. And lignin, because of its phenolic nature, absorbs that harmful ultraviolet to protect the plant and keep it healthy. So the way we convert biomass, the plant material that we generate on a lot of these plant farms, whether it's corn or grasses or even trees, is we have to deconstruct that cellulose, that cellulosic biomass, to extract the cellulose away from the lignin so we can convert the cellulose to ethanol. It turns out to be a horrible process. It hasn't changed much since we pulped paper over a century ago. So what would you do to be able to get more cellulose out of wood? Well, some people thought maybe we could knock down the genes for lignin. If we didn't have lignin, maybe you get a lot more cellulose, the problem would be a lot simpler, and we could extend our fuel supply. So that's in fact what the early geneticists did in first-generation plant feedstocks about 20 years ago. And what they found was that we could take plants that had lower levels of lignin, and if we treated them with enzymes that I know something about and some other chemicals, we would be able to get more cellulose than we could out of normal wood. I should say more accurately, we could get cellulose out more efficiently. But it turned out there were some drawbacks. First of all, though it was more efficient to get cellulose, we actually got less material because there was less cellulose there to start with. So one end product of manipulating the lignin levels was that we messed up the metabolism of these plants and they weren't making enough cellulose anymore. The other problem was if you grew these plants to maturity, they tended to wilt. They no longer had that structural strength that normal plants would have. And then lastly, they were a lot more sensitive to environmental stresses like ultraviolet light, but also other stresses like high salinity, if it was salty water, temperature, things along that nature. So these were interesting challenges, and I thought, well, gee, what could I contribute to this? So this is my one homage to the more artistic inclined who were speaking tonight. This is a pic. Oh, sorry, I went too fast there. This is a photo here of a poplar plant. Our lab works on poplar trees that's grown in the laboratory or in the greenhouse, and we can put that plant outside, and it grows quite normally. This is a small plant, but it'll grow to maturity over a course of 10 years. It'll grow nice and tall, like a lot of poplar plants in your neighborhood or maybe at your home. This is a poplar plant that has a lower level of lignin that's currently being evaluated as a biofuel feedstock. So I might say it has a little bit of a different feature, wouldn't you say? Almost has a beautiful magenta hue to it. Well, the reason it does is because this plant, when it's outside, it can't cope with that ultraviolet light. So the plant responds the best way it can. It stimulates the synthesis of other compounds to protect it from the sun's ultraviolet light. In this case, it turns out to be the same compounds that people find in orchids that give them the remarkable color. Okay, so a lovely magenta color. It's an anthocyanin. So when I saw this in one of my colleagues' lab, I thought, gee, we could manipulate the molecules, the pathways, the metabolism in plants, and we could find some of these outcomes, and we could see if we could utilize some of them, some of the good ones, in poorly performing biofuel plants, and maybe eliminate some of the negative ones, and see if we couldn't generate a new version of poplar plants that might be more effective feedstocks for biofuel. So because this is so important, JD said I could have the next hour to tell you all about our current results. But because we're running short, okay, and he gave me the hook, I'm not going to do that. I'm just going to summarize very quickly. So we're looking at three key aspects about these plants, the same three shortfalls that the original first-generation poplar plants had. One is we're very interested in trying to see if we can adapt their stress response. That is how they respond to unfavorable conditions like growing on non-ideal land, not like the way we plant our food crops. But if we can adapt some of those processes or pathways to make more effective responses to less hospitable conditions. So we're trying to get the good parts of those responses and incorporate them into novel plants and eliminate the bad responses. The other thing is we're trying to address the issue of metabolism. So one of the key metabolites we focused on is nitrogen. As you all know, nitrogen is lacking in the soil unless there's decaying organic matter. And plants need nitrogen in order to grow. One of the reasons why we put so much of it on all of our crops. Well, plants, and in particular trees, are pretty clever about how they manage their nitrogen. So they'll store their nitrogen in their bark in the winter. And in the spring, they'll utilize that stored nitrogen from a sink, is what we refer to it as, to provide the nitrogen that it needs for new growth until it exploits it, it uses all that up, and only then will it make the molecules and the transporters and the other compounds it needs in the roots to extract nitrogen from the environment. So we're manipulating those sensors and those sinks to be able to see if we can extend that nitrogen, internal nitrogen supply in plants, so that we might be able to prevent adding nitrogen in fertilizer in order to get good growth. And that has ramifications beyond biofuel. Obviously, it has ramifications in crops as well. The last thing, and I realized I neglected to mention this, one of the downsides of these plants with low lignin, lignin provides a great barrier to pests and disease, microbes, fungi, bacteria, as well as insects that bore into plants. Because of that, a lot of low lignin plants tend to be very sensitive to disease. And so we're looking at ways, first of all, that plants get infected by some fungi, some of the most important fungi that infect poplar, and then trying to see if we can't engineer their immune system. So now plants don't have antibodies like we do, but instead what they do is they have molecules that recognize pathogens. And so what we're doing is modifying those molecules to see if we can't do two things: expand the spectrum of pathogens that a plant might recognize, and then let them recognize those pathogens at lower concentrations. So that way we can stop disease before it starts, a lot like a lot of us take a botanical dietary supplement to stay well, rather before we get disease, which we alluded to earlier today. All right, so there are two things that I want to leave you with as a take-home. One is, as I mentioned earlier, this work was critically dependent on a vast network of people, some of which are listed on this slide, and I want to mention a couple things. So a lot of you might have this conception of a scientist huddled over a bunch of papers or a computer coming up with a new model or trying to assimilate some data from a colleague or something along those lines, and that happens. But science has become a social endeavor, big time, okay? To solve a problem like biofuel, I have to reach out to people who know about land management, who know something about crops, who know about plants. I don't know anything about that. I know about molecules and pathways and how to manipulate them, how to engineer them. But we all have to work together. And at the university, we try to develop what we call T-shaped students. They have a lot of depth in one area, but they have an ability to reach out, interact with other people who also have depth in other areas. And that's exactly the kind of network that we've developed to tackle some of these questions. It's the kind of thing that we're doing now. We also engage the public quite a bit because of the tremendous importance. You people are the stakeholders. And I want to mention that some of the caretakers for hard-earned taxpayer dollars are agencies like the Department of Energy that generously support us. The last thing I want to leave you with is one thing, or you need to know three things, I should say, about biotechnology. It's really simple. It's complicated, it's expensive, and it's unpredictable. Aside from that, we got it down, all right? So the thing about that is that we're at the early stages. We're trying to understand mechanism in these plants, in these complex biological systems, so we can learn enough so we can engineer their properties in some predictable outcome. Now think about it. Think about what could be. If 25 years ago we had this DASER meeting, and I said to you, each and every one of you in 25 years would have a computer at your house more powerful than any Department of Defense computer that's supposed to protect our nation. And maybe you didn't just have one per family, maybe everybody in your family would have one, and maybe even kids in high school or elementary school, and you changed it every 2 years for a better model. This is what we have. So now think forward 20 years, okay? What we're going to be able to do in biosystems as we learn enough about the mechanism to manipulate them and to engineer them for some predictable outcome. So they'll be safe, so they'll function the way we want to, and they can help us advance the goals of society. So thanks very much.